Hello, my name's Mrs. Tyler and we're here today to do a bit of GCSE biology revision. Today's content is on decay, which is only on the triple GCSE or the separate science GCSE. So unless you're doing triple or separate science, you don't need to do this. If you are doing triple, then welcome, you're in the right place. We're going to make something that looks a bit like this today. So you can see we're looking at the carbon cycle, we're applying it to rates of decay, and then the required practical looking at the effect of temperature on enzymatic breakdown of fats and milk. And lastly, we'll be looking at biogas production. Right, let's get started. So I'm going to put the title in the middle, as always, and today we are thinking about decay or decomposition, rotting. We're going to divide the paper in half. And then this half, I'm going to divide into about 1/3 and 2/3. A smaller section at the top. That's actually more like 1/4 and 3/4, isn't it? And this one, I'm going to divide in half that way. So 2 even-sized sections there. In this one, I'm going to recap the carbon cycle. In this one, I'm going to recap the rate of decay and factors that affect it. In this one, we're going to be looking at biogas. And in this one, we're going to be looking at the required practical, where you look at the rate of decay. It's a bit of a crazy one, because really, you're looking at the effect of temperature on lipase in milk. So all of this is on triple only. So if you're doing dual award, you don't need to do this. Let's start off then with the carbon cycle. We're going to draw one out. So we're going to start off with carbon dioxide in the atmosphere. And you know that the amount of carbon dioxide in the atmosphere varies, but it's very, very low, about 0.03, 0.04%. Here, then, I'm going to draw a tree. And hopefully you know that trees and other plants or producers take in carbon dioxide from the air in the process of photosynthesis. And turn it into glucose, sugar. So carbon dioxide is taken out of the air by photosynthesis and turned into glucose. You also probably know that plants also use that glucose and break it down to release energy and make carbon dioxide that goes back up. That's the process of respiration. Animals, on the other hand, get their carbon by eating it. So I'm going to draw a cow. There's the cow's face. I'm going to draw 2 horns. Like that. Cow's going to be eating some grass. Draw the cow nose there, like that. I'm going to write cow, just in case you can't tell it's a cow. Okay, so cows and all of the herbivores get their carbon by eating it, and eating it in the form of plants. So the carbon gets passed from plants to animals by the process of eating or consumption. Now, Some of that carbon dioxide, or a lot of it, will be respired. A lot of the carbon in the cow will be respired and returned to the air via respiration. Some of it will turn into cow body mass. Some of it will be excreted in feces or waste. And really, delightfully enough, what we're thinking about today is what happens to that feces and what happens to the biomass in the cow when the cow dies. So I'm going to draw an upside down cow now, really badly. There's my dead cow. And so what happens to the excreta, the fecal waste, and the dead material is it decays. So both of these undergo the process of decay. And that really is what we are thinking about in today's lesson. Decay then happens because of decomposers, types of bacteria and fungi that live in the soil, breaking down the protein and the carbohydrate and the fat that are in that dead stuff and releasing it either as minerals back into the soil or respiring themselves and putting it back into the air as carbon dioxide. So these are our fungi and bacteria, our decomposers, which are bacteria and fungi. And when something dies or is excreted as feces, they secrete enzymes that chop it up. And they will use some of that for energy, just like plants and animals do, via respiration. So some of the carbon will go back into the air. But also, what they do is a very useful thing, which is release minerals back into the soil. And that means that plants can take those minerals up and turn them into proteins, for example, and it can cycle back round this way. So decay replaces minerals back into the soil. So, for example, nitrates and magnesium ions will go back into the soil, and they're then in the soil, available for this tree to take up again there. So like I've said, decay is actually really useful, replacing minerals back into the soil. Can also be annoying because it means that if it's fresh food, if it's meat that we want to eat, for example, or vegetation that we want to eat, we don't want it to decay. We want to keep it fresh. So the rate of decay is important. And whenever we're thinking about the rate of decay, we need to remember that decay happens because of bacteria and fungi. And they cause decay by secreting enzymes. So factors that are affected or affect the rate of decay are primarily, number one, temperature. So just like with enzyme theory, when it's warm, there's more kinetic energy, there'll be more reactions, which means the rate of decay will be faster. And if it's cold, there's less kinetic energy, so there'll be fewer reactions, so the rate of decay will be slower. We also need to remember that if it gets very hot, then the enzymes will be denatured, and so there will be no decay at all. So warmer leads to faster decay. until it's denatured. That will depend on the enzyme and the environment in which that enzyme's in. And equally, when it's colder, it will be slower decay because there's less kinetic energy. The bacteria and fungi often are aerobic, which means they often need oxygen. So a lot of the time, oxygen affects the rate of decay. And that's because it affects the rate of aerobic respiration by these bacteria and fungi. Needed for aerobic respiration. Also, things like the amount of decomposers, the amount of fungi and bacteria, the more bacteria there are, the faster the rate of decay. So the number of bacteria and fungi. So more bacteria is to more decay. And lastly, the amount of water, or the presence of water. And that, again, is because the bacteria, the fungi, need water. So whenever we're thinking about the rate of decay, we're thinking about factors that are going to affect the rate of growth of bacteria. One use of decay, then, that you need to know about is in the production of biogas. So we can use this decay process where the bacteria and fungi break down organic material, so fecal matter here, into carbon dioxide and minerals. If you have anaerobic bacteria, however, they don't make carbon dioxide, they can release methane. So making biogas is using anaerobic bacteria, an anaerobic. So anaerobic bacteria to break down the waste and produce methane. And it's the methane that we want. So it needs to be, that's not very clear, anaerobic conditions. So let's have a think about how that can happen then. So to make biogas, we need anaerobic conditions, so there needs to be no oxygen. We need a source of bacteria, and we need a way of collecting the gas. So biogas generators are often submerged. The source of waste can be from animal or human. I'm going to draw a toilet here. So this is my waste pipe. This is the surface here. And I'm going to draw a toilet directly above it. So one efficient way of generating biogas is by using human waste. So you attach the toilet directly to your biogas generator, but it could equally be animal waste. So you could have a barn and you could have a way of getting the waste from the barn down into the biogas generator. So the source of waste can be human, animal, or vegetable, or plant-based. Whatever the source of waste is, it needs to go down into a biogas generator. So I'm going to draw the big pit here. I'm going to draw another pipe coming off the side there. This chamber is going to be our biogas generator. It needs to be sealed in because it needs to be anaerobic, and they tend to have a domed top whereby there's a pipe coming off the top and that is where you collect the gas. Inside here then is a heady mix, as you might imagine, of waste materials, but you also need those precious anaerobic bacteria in there, fungi, and they will release enzymes, break down the waste material, and release methane gas that will bubble up to the surface. So the methane will gather up here, and then it can be collected out of this pipe here. So this is the chamber where anaerobic bacteria break down. In this case it's feces, but it could be any other waste. You've also got an extra byproduct on here, a useful byproduct. When all the useful stuff has been broken down by the bacteria and you've got all the methane off that you can, then we've got left lots of mineral ions in here, just like the bacteria and fungi put mineral ions back into the soil. The mineral ions will be free in here, and so the slurry is a great fertiliser. So you need an outlet pipe for the slurry, which contains mineral ions to use as fertiliser. And that's one use of decay. On to our last section then, where we're going to be looking at the effect of temperature on the rate of decay in milk. So I'm going to draw 2 tubes. I'm going to leave a bit of a gap at the top because I'm going to draw something in there afterwards. So 2 tubes, test tube 1 and test tube 2. And in this experiment, we start off, we put milk in, that's our source of fat. We make it alkaline, so we add bicarbonate. And we add an indicator, which is phenolphthalein. Can't spell it. Phenolphthalein, something like that. And phenolphthalein, when the bicarbonate ions are present in milk, should be pink. So this should be pink, which means it's alkali. And the reason it's pink is because it's got alkali ions in, and in the milk, the fat, or the lipid, hasn't been broken down. So remember, fat is a backbone of glycerol, and it's made out of 3 fatty acids, and they're all still attached to each other. So the glycerol is attached to 3 fatty acids. However, when we add lipase into this reaction, so lipase ends in ase, which means it's an enzyme, it breaks down that fat and it snaps off the fatty acid chains from the glycerol. So they're now free in solution. So now we've got fatty acids on their own. And fatty acids, as the name suggests, are acidic. So as the lipase chops up the fat, it makes more and more acidic molecules, which means it gets more and more acidic, which means eventually this pink indicator showing it's alkali eventually goes white. In fact, it goes clear really, but the milk is white, which means it is neutral. The fatty acids neutralize the bicarb, the alkali that we put in the start. So it stays white this time. You've still got milk, bicarb, indicator. You've now got lipase as well. And so it goes white. So in this experiment, we're going to time how long it takes for this indicator to stop being pink and go white. In this experiment, we're changing the temperature because we're looking at the effect of temperature. So the independent variable, the thing that we change in this one, is temperature. So I'd set up one tube in icy water at 4 degrees, one tube at room temperature 20 degrees, one tube at 40 degrees, one tube at 60 degrees, one tube at 80 degrees. I'd change the temperature. The dependent variable, that's the thing that we're measuring, is, sorry, I forgot how to spell measuring, time taken. So we're measuring time. We'll do that in seconds. The control variables then are the things that we need to keep the same each time. So the control variables, in other words, the things that make it valid, remember an experiment to be valid needs controls, then we would need the same volume of milk or lipase or bicarb, or the same concentration. Notice we're not saying same amount, okay? Now, if I want to make this experiment more accurate, I will need to repeat it at each temperature 3 times and calculate a mean. So if I want to make it accurate, repeat it at each temperature 3 times and calculate a mean. And that calculating a mean is important in exams. Once I've got the time it takes from it to be pink to being white, then I can calculate the rate by putting 1 divided by the time taken. We say 1 because 1 denotes a colour change. There's no numerical value to this colour change. It's either pink or white. So we say it's not there, it is there. So that has a value of 1. If those numbers were super small, I could do something like 1,000 divided by 1 to make the numbers a bit easier. Sorry, 1,000 divided by time. And improvements we could make to this method. We could try smaller intervals of temperature, so do more temperatures with smaller intervals so that we can find the optimum temperature a bit more accurately. Or we could do something else like change the pH. Okay, so that's our summary of decay. This is particularly important for a higher triple. It's more likely to come up. Your job now, you know it, is to read, cover, recite, and then go and have a go at some exam questions. And I'll see you next time.